We have guests.
I don't remember when Tom started with it. I can't remember a world in which Tom was not in it. Okay, I mean, it's 40 years. We've been here 50, so he started 10 after we came.
I've been in the lab, some 28 years, and Tom joined the lab before I did. So for all these years, Tom has been an everyday part of our lab's life in every capacity.
My job at Stanford is in research and development electronics engineer. When they first hired me in the Herzenberg laboratory, I was hired to interface the cell sorter, the prototype cell sorter, onto a computer. They didn't have any computers then. Fax 1 it was called. There was only 2 of them in existence at that time. One of them went to NIH, and the other one was here.
Led Herzenberg's group actually commercialized flow cytometry, which has been an incredible tool for medicine, for research, where you would have a limited sample and be able to get very little information. Now we can get an incredible amount of information from that single cell via the use of flow cytometry. So for its day, it revolutionized the field.
Well, the cell sorter was mainly a research instrument which would allow people to do experiments with individual cells that they couldn't do any other way. The alternative was put them on a slide under a microscope and pick them out with a needle.
It started out with a very simple, straightforward focus, either looking at DNA content to try and cure cancer, or to look at the surface molecules of cells of the immune system. And that has just mushroomed into so many different areas.
This is what we count CD4 T cells in HIV patients when we're monitoring the progress of the disease. This is what we look at leukemias to tell what kind of leukemia it is so we can decide which kind of chemotherapy.
It's very important in bioengineering research where people are trying to make synthetic organs.
Lung samples where we want to look in cystic fibrosis at what kind of cells are there.
Neurological research looking at brain cells.
T cells, B cells, thymocytes, different types of white blood cells were all sorted out by fax.
Tom's skills were invaluable in making a lot of these things happen that we worked on to enhance the cell sorting.
He was an essential part of our whole cell sorter program. Probably wouldn't have been the success that it was without Tom.
I guess when I was small, I always thought I'd be a professor or something, you know, making inventions and things. That was something I thought about. I played with batteries and stuff and, you know, lighting light bulbs and making crystal sets to receive signals, radio signals, and I made it all from scratch. And so that was my first experience at electronics, I guess. Maybe that's why I got into this field, because I really enjoyed that kind of thing. After 4 years old, I remember everything. Getting spanked by the kindergarten teacher or whatever, I remember all those things. It was Mrs. Landell's.
His memory is encyclopedic. You look in his office and it's got box after box after box, and you think in the chaos he can never find anything. You ask for something, in 2 minutes he'll go to the very back underneath through things and he'll find exactly the part he needs.
Whenever we need something, they ask me, do you have this? And I know where it is, so we can fix anything that's in our cell sorter facility.
Tom's been a great troubleshooter for everything over all these years, and he's built a lot of the equipment, and he's kept a lot of the pieces in his little office there.
Just trying to get to talk to him. He was such a notorious pack rat that his office was so full that there wasn't any room for him anymore to sit. If you wanted to talk to him, you'd knock on the door, and then you'd wait for about a minute while there was just sounds of shuffling inside, and the door would open 3 inches, and he said, yeah, what do you want?
If you see how our group is, you'll see that every one of them is talented in some way in their field. And it's nice to work with people like that. The group that we have today is the same group that we had when we first started. We're like a family. I don't know, a family that's stuck together all these years, so we know each other very well after knowing, you know, 35 or 40 years working together. I think that's kind of unique, but it's the only job I've ever had. I've never worked anywhere else.
We all value him immensely, as do Lee and Len. I mean, there's just no question. We wouldn't have had the instruments without Tom.
Every lab should have more than one Tom. He was just the solid engineer who went all the way through this and ultimately built the designs for all the modern machines.
It's very satisfying to do something unique. It's nice to go and see that the whole world is making use of this, and they're making discoveries and studying the immune system and probably curing diseases. And without this cell sorter, they wouldn't have been able to do this. I'm proud of that. It's cutting-edge science. And so it's very satisfying to be part of the research team that did this.